Hello.
Hello, everyone, and thank you all for joining us during the Lytham Partners Fall 2023 Investor Conference. My name is Robert Blum, Managing Partner of Lytham Partners. During this webcast, we welcome Kiora Pharmaceuticals, ticker symbol of KPRX on the NASDAQ. Joining us today from the company is Melissa Tosca, Executive Vice President of Finance. Today, I've asked Melissa to run through the company's slide presentation, and then we will engage in a short question and answer session at the end here. Before I do turn it over, I want to remind everyone that management is available for one-on-one meetings later today. If you've not already signed up for one-on-one and would like to do so, you can send me an email. That's Blum, B-L-U-M, at lythampartners.com, or visit the website, lythampartners.com/virtual. Click on the one-on-one meeting request button, and we will look to get you taken care of. So with that said, Melissa, let me turn the floor over to you to walk through the presentation.
Awesome. Thank you, Robert. Hello, everybody. It is my pleasure to introduce Kiora Pharmaceuticals. I'm Melissa Tosca, the Executive Vice President of Finance for Kiora, and I look forward to sharing more about our company with you. One housekeeping item before we get started. As a public company, we're required to include a disclosure regarding the forward-looking nature of some of the statements in this presentation. Simply put, the materials are based on our current expectations and as such may be subject to change. So let's jump in. As you know, there are many ophthalmology companies today. Kiora is unique, however, in that we are focused on orphan retinal disease indications where there is significant unmet need. We seek to improve sight in patients with severe vision loss due to inherited or age-related diseases. This is a significant market opportunity, and we are well positioned given our expertise. Although we're a small company, we have extensive experience in retinal disease and drug development, so we're focused where we know we can be most successful. Vision is one of the most precious senses. For someone who's losing their sight, they are overcome with fear and often despair. How will they navigate their day-to-day life? What will they miss out on? How will this impact their families? I witnessed this despair firsthand as a child when my grandfather lost his vision. He lived the last 20 years of his life blind, and sadly, his very active lifestyle completely changed due to the loss of his sight. Prior to losing his vision, my grandfather was an avid outdoorsman. He loved to go camping and fishing especially. However, as you can imagine, he became much more confined to his house once he couldn't see. The graphs on this slide highlight that blindness is ranked as one of the worst conditions with the greatest effect on day-to-day life, higher than many horrible diseases, including Alzheimer's and even cancer. At Kiora, we are focused on retinal disease therapies to address these fears and to help give back what's been lost. Our pipeline is comprised of orphan disease indications. For our lead asset, KIO-301, we are using a small molecule photo switch to confer light sensitivity and have started programs in retinitis pigmentosa, choroideremia, and Stargardt's disease. We have also a small molecule DHODH inhibitor and significant data already generated for our KIO-104 program for posterior non-infectious uveitis. Over the next 12 to 18 months, we have many clinical and regulatory milestones. As you can see, there are several value inflection points ahead, including both proof of concept and top-line data readouts for multiple indications. This is truly an exciting time to be at Kiora and a great opportunities for investors given the growth trajectory that's ahead. Let's talk about KIO-301, our small molecule photo switch. This is a simplistic depiction of the retina. At the far right, you can see the rods and cones, which are the photoreceptor cells that take light and confer it to electrical energy. That energy is transmitted to bipolar and retinal ganglion cells called RGCs, which relay the signal to the brain through the optical nerve. In many retinal diseases, the rods and cones die, causing blindness. However, since RGCs remain alive, we're able to target those cells and turn them into light-sensitive cells. Our small molecule KIO-301 goes into RGCs through specific areas of degeneration and travels to specific ion channels. In the presence of light, its linear form changes to a perpendicular shape, which physically blocks the ion channel from outflow and which enables the cell signal that light is present. When light goes away, the molecule reverts to its linear state and the signal turns off. Retinitis pigmentosa is our first disease indication. There are currently no drugs approved in retinitis pigmentosa, and patients generally begin losing their vision in their upper teens to 20s, eventually becoming legally blind in their 50s. RP is a group of diseases that can be caused by mutations in well over 50 genes. With over 100,000 patients in the US, this provides a significant market opportunity. We just completed our KIO 301 phase 1b abacus study. This was a first in-human study where we looked at different drug amounts among patients with different levels of disease progression. All of the patients were considered off-chart, meaning they can't read even the first letter of an eye chart. We had a host of efficacy endpoints, and of course, safety was of paramount importance. We are very excited to be releasing full study data at the American Academy of Ophthalmology conference in November. While it is essential to have patients complete objective and measured testing, it's extremely encouraging and inspiring to hear subjective feedback as well. So on that note, I have a few short patient videos which highlight some of their experience that I'll play for you now.
Later that week, I went back home to Melbourne, and one of my hobbies is bike riding. And we go out bike riding early in the morning, often leave when it's first dawn or dark. And as part of that, I put the lights on the bike, and and on the tandem bike, I've always relied on someone else to say, is the light on? Is it flashing? And because I had no concept myself, and I sort of looked at this light and turned it on and go, hold up, I can see that light, and I can see whether it's on or whether it's flashing. And I think that that was the first time, not only in a clinical sense, but actually it really goes, wow, this is really useful.
Last night when I got home, because I was in the city at Kiora yesterday doing some tests, and I've got this device on my bedside, and it's got these two little lights, and I can never see them.
Yes.
And I saw them last night.
Okay.
And I went, wow.
I think that was very impressive at the foyer where you come into my unit. It's quite a small area, but again, I thought the light was getting dimmer and dimmer, and I'd have to look up and find to see whether I could see the light. Is it turned on even? And then when I said, I thought, it needs to be made stronger. Well, again, 3 days after the injection, it was like the whole area is floodlit. The light was, without me having to look, I could see the light in the ceiling. And it was just like the whole little area is floodlit, and I could even see from the kitchen, I could see the glow from that foyer area.
This is a case study from the trial, which highlights some of the functional MRI data and provides both objective and subjective data. Specifically, within the region of the brain that supports visual processing, we saw improvements in the ability to perceive light, as well as the quality of life. In terms of next steps, we're looking forward to sharing full data this quarter, as mentioned. Additionally, based on our initial findings, we received approval to increase our enrollment with 3 additional NLP, no light perception patients, since our study initially only had 1 NLP patient. We plan to request a preliminary IND meeting with the FDA before year end, and will be initiating a sham-controlled phase 2 trial in early next year. Lastly, given the outcome of our first study in RP, we are very excited to expand into other inherited retinal diseases with similar pathology, specifically choroideremia and Stargardt's disease. Choroideremia, like RP, is an inherited retinal disease that leads to blindness. We recently received approval to expand our current trial to treat patients with choroideremia. We also recently announced a partnership with the Choroideremia Research Foundation, which is the world-leading nonprofit patient advocacy group. We are benefiting from their specialized educational resources, as well as access to their key thought leaders. In addition, Through their network, we're gaining assistance in finding patients for our trials and collaborating on the design of our protocols. Stargardt's disease is another inherited retinal disease with unmet need that we believe has application for our KIO-301 small molecule. We're planning a similar clinical design targeting the same locations as our phase 2 Abacus trial, which will provide us with synergies, and we believe that will translate into cost savings as well. So while we're wrapping up our current phase 1b study, we've kicked off our phase 2 study preparations. This will be a larger study comprised of 3 cohorts and will include a sham injection. We'll begin the trial in early 2024 and expect to have data in early 2025. Moving on to our other intravitreal asset, KIO-104 is a novel non-steroidal small molecule, which is a DHODH inhibitor. This enzyme inhibition has been shown to effectively treat autoimmune diseases and is a multi-billion-dollar market for Sanofi and others already. We have a best and first-in-class ophthalmology drug, which provides a significant advantage, and we see this as a great market opportunity. Non-infectious uveitis is an area where we believe KIO-104 could help improve intraocular inflammation. We're taking a steroid-sparing approach to treat chronic inflammation that has generally been treated by steroids. Steroids, we know, can cause adverse events leading to cataracts and glaucoma, so there's a great need for an alternative non-steroidal therapy. We previously completed a phase 1 study in uveitis, which showed exciting data in both vision and edema improvement. So we believe there is significant unmet need in this area. I'll wrap up with a brief corporate overview. We are a team of experienced biotech executives. We bring ophthalmology expertise as well as drug development experience and have worked across all asset life cycles from development to commercialization. We have an outstanding board of directors who bring diverse backgrounds, expertise, and provide great oversight. And lastly, we are supported by an amazing scientific advisory board comprised of the world's thought leaders in ophthalmology. Thank you so much for learning about Kiora, and I look forward to addressing any questions. Robert, I'll kick it back over to you.
Thank you very much, Melissa, for that overview here. And yeah, just a couple of topics maybe I want you to expand on if you could. You know, which patients do you feel will benefit the most from KIO-301?
Sure, yeah. I mean, we're starting with off-chart patients and seeing some very promising results. So there's certainly a possibility that patients with earlier stage disease will also benefit. However, based on the way KIO-301 works, patients will need to have some vision loss. So including, peripheral vision loss really for the drug to have a likely benefit.
All right, Melissa, where do you see KIO-301 fitting, sort of intervening in the course of treatment relative to gene therapy?
Yeah, so gene therapies are mostly targeting earlier stage patients where they aim to slow or prevent further progression. We're focused on later stage patients where we believe we can restore lost vision.
All right, and then so what are some of the advantages of small molecules to gene therapy?
So commercially, there's greater ease of delivery and more pricing flexibility that's associated with small molecules. And as a result, there's favorable reimbursement that can drive patient access. Additionally, most gene therapies target a single mutation, and since there's more than 150 unique mutations, that can cause RP or many other IRDs for that matter to really undergo a long delay from the time the gene therapies are developed to when they can actually reach all of those mutations, if ever. So KIO-301 can be used in patients with any of the gene mutations. This is referred to as it being gene mutation agnostic. And long term, we can envision the use of KIO-301 in combination with certain gene therapies at a stage where it may still warrant slowing the disease progression with the gene therapy, but then we can also then restore any lost vision through the utility of KIO-301.
Maybe talk about some of the advantages of a small molecule to gene therapy in general there.
Sure, yeah, absolutely. I mean, gene therapies are mostly targeted in earlier stage patients, so they're aiming to slow or prevent further disease progression. We're really focused on later stage patients where we believe we can restore vision. So that's really the difference between the two.
Okay. You touched on some of the sort of the upcoming milestones here as it relates to Kio 301, but maybe just sort of walk us through kind of the sequence of next steps in bringing Kio 301 to the market here.
Sure. Yeah, we are preparing a phase 2 clinical trial, which I highlighted, and that'll really allow us to further explore the dosing and inform us really in a design capacity as we prepare for the registration clinical trials. So this will be run throughout 2024, and if all goes well, we plan to kick off the later stage clinical trials in mid 2025 in the US and hopefully in Europe as well.
Okay, perfect. Sort of taking a step back here, have there been any notable success stories in the treatment of inherited retinal disease?
There have. So Luxturna, Luxturna, if I'm saying that correctly, is probably the best example. It's a gene therapy that was approved for a single gene mutation that causes a subtype of Leber congenital amaurosis. And the product was developed by a company called Spark Therapeutics, which was ultimately acquired by Roche for not a small dollar amount of about $5 billion.
Got it. Okay, that's helpful. Sort of, what are maybe some of the other notable therapeutic developments in the ophthalmic industry right now?
Yeah, so this year there's been a lot of buzz around FDA approvals of 2 drugs for geographic atrophy, which geographic atrophy is a late stage form of age-related macular degeneration, and that can lead to vision loss. So these drugs were developed by Apellis and Iveric, with Iveric having been recently acquired by a Japanese pharma company called Astellas, and that acquisition was for about $6 billion.
Got it. Okay. Last question here, and I think you touched a little bit on this, but what's sort of the long-term opportunity for Kio 301 beyond RP, right?
Yeah, so I mean, looking at the pathology and the progression of inherited retinal diseases, there's several that result in the death of photoreceptors, while the RGCs, which are our target cells, remain alive. This includes choroideremia, which we talked about, and Stargardt's disease as well. So inherited retinal diseases is really like our focus area, and that in the U.S., the healthcare system has a spend of about $32 billion a year for inherited retinal diseases. So drugs like Kio 301, they have the potential to make a considerable dent in that healthcare costs by improving patients' vision. And yeah, that's where we're really excited to be.
Fantastic. Well, we'll leave it there. Melissa, thank you very much for your time today and for sort of walking us through the presentation. Greatly appreciate it. Again, I just want to remind any of the listeners out there, if you haven't signed up for a one-on-one meeting already and would like to do so, shoot me an email, bloom@lythampartners.com. Go to the website, lythampartners.com/virtual. Click on the one-on-one meeting request button, and we'll look to get you taken care of. So, Melissa, thank you very much for your time today. Greatly appreciate it. Hope you enjoy the conference.
Thank you so much.